We're now going to be looking at how our immune system can recognize self from non self. So, our learning intentions for today is to be able to understand that when a pathogen enters into our body, it causes physical or chemical changes that stimulate the immune responses in the host. So, um, we're first going to talk about our major histocompatibility complex here. So, what this is let me get a pen instead, shall I? So, imagine we've got I'm going to use the circle here, a cell, and that's the nucleus. And this is one of our cells. It's not very good, but we've got a little major histocompatibility complex, so that's the MHC here. And so these are protein markers on the surface of our cells. So they code for a very specific set of genes that are unique for all individuals, and they help us recognize foreign molecules. Okay, so what happens is that we might have, let me go in different colors so it's a bit more obvious. So if we have a foreign particle, and on this foreign particle is something called antigens. Antigens, and that's these little spikes here, right? So what happens is that an MHC might come into contact with um, one of these antigens, and it will recognize it as being non-self. So what might happen is that the this kind of pathogen or foreign molecule gets consumed, and then what happens is that our major histocompatibility complex. Oh, let me find that same color I had. So we got another one over here. I'm going to draw it very nicely. There we go. We'll then display um, the antigen that is non-self. So it actually highlights itself as being something that needs to be destroyed. Okay, so it's actually these antigens here um, that are either going to be considered self or non-self. So again, if I get a different color, oh, this is a highlighter. I don't want a highlighter. Um, so if we have another cell, and this is our self, and again, our self has our own antigens, right? And we might call these self antigens. Okay, so this one may come across our cell, and our cell recognizes it, so not much more needs to be done anymore. Okay, so my drawings, I'm going to be honest, probably aren't the best drawings. So instead, let's have a better look at this here. So we can see that we've got our normal human cell here, and we have an antigen that's come in, and what you can see here is the process of our cell or this foreign particle being eaten is called phagocytosis, and it is now presenting um, our antigens here by the MHC complex. So it's presenting that there is something that's not quite right. And so we will go into further detail on this in another video, but it's pretty much flagging something's not quite right in our immune system. There is, or in our own body, there's something that might need getting rid of. So to make it a bit more clear, an antigen is a marker that tells your immune system whether something in your body is or is not harmful. So, like I said, we do have antigens, or our own body has antigens unique to us and our own cells, and our immune system uses that to say, okay, I recognize that antigen, I'm not going to attack it. Antigens exist on things such as viruses, bacteria, allergens, parasite, proteins, tumor cells, like I said, normal cells, and even like toxins and chemicals and all of that stuff. All of that has antigens that can be recognized by the immune system. So each has a unique shape, as I said before, that your immune system reads like a name tag so they know whether it belongs to your body. So this means that your body recognizes your own cell as you, but other antigens as intruders. So remember when we had COVID and we had to take the rapid antigen tests? Essentially, a rapid antigen test is actually trying to detect the antigens that are specific to COVID 19. So that's why if you're sick, it's not going to go positive. It's only going to go positive if it's detecting the antigens from COVID. So that's why there was a bit of a discussion about how, well, with all of these new mutations and variations of COVID arising, they were worried that the rats wouldn't work for it just because it might be that the antigen's a little bit different, so it might not detect it as well. So, just to have a bit of a look at some of the implications of antigens and things like that, we've got something here. It's called a rhesus factor. So, people, you know how you might have like you're an AB positive or an AB minus in terms of blood type. Well, this rhesus factor, you either do have it or you don't. So that's why you've got the A positive or A minus when it comes to our blood types. So this is inherited from either parent. Okay. So if the mother has a negative blood type, so I don't know, O minus, that means they don't produce this rhesus antigen. So this can cause complications during the second pregnancy, in that what can happen is that the mother actually might start producing antibodies to attack the second babies, or the baby who has a blood type that's not familiar to the human, to the mother's immune system. It actually might start attacking the baby, which can definitely cause a few complications. But can we can get around it by using medication or treatments to try and stop this from occurring.